AZLAN ADNAN MA presents The Coconut's Secret Journey, Origins and Genetics, Wednesday, 22 July 2026.
For generations, scientists believed they had pieced together the story of one of the world's most iconic tropical plants. Yet groundbreaking genetic evidence is rewriting that narrative. Ancient coconut DNA, analyzed through cutting-edge genomic techniques, now reveals an extraordinary saga stretching back thousands of years. A tale of survival against pounding waves and salt spray, human ingenuity on the high seas, and the quiet power of a single species to reshape landscapes, cuisines, and cultures across the globe. Imagine standing on a sun-drenched coastline in island Southeast Asia, where the air hums with the rustle of broad palm fronds and carries the sweet, earthy scent of fallen husks warming in the sand. From these vibrant shores to forgotten ancient trade routes etched across vast oceans, the coconut's true history proves far more complex and captivating than anyone once imagined. Where did coconuts really come from? How did their hard, buoyant fruits cross seemingly endless blue expanses to colonize distant shores? And what role did early civilizations play in their remarkable spread? Today, modern science, drawing on advanced genomics, archaeology, evolutionary biology, and linguistic clues, offers compelling answers that bridge deep time with the intimate rhythms of human adventure. Two ancient ocean basin lineages. For decades, the coconut, Cocos nucifera, seemed to possess a murky or scattered origin, its wild ancestors lost in the mists of prehistory, and its presence across so many tropical regions explained vaguely by buoyant fruits drifting lazily on ocean currents. DNA analyses of thousands of coconut accessions worldwide have brought clarity, identifying two major, highly distinct populations that tell a story of parallel journeys rather than a single, simple path. The Pacific group traces its roots to the lush islands and coasts of maritime Southeast Asia, particularly the rich biodiversity hotspots of the Philippines, Indonesia, and Malaysia. Here, amid the humid embrace of tropical rains and the rhythmic crash of waves on coral shores, early peoples recognized the coconut's potential and shaped it through careful selection. This group displays unmistakable signs of domestication, the emergence of compact dwarf varieties, self-pollinating traits that ensure reliable harvests even in isolation, and the rounded neovi fruits optimized for human use, with a higher ratio of sweet, milky endosperm to tough, fibrous husk. Crack one open under the blazing sun, and the cool, refreshing liquid inside offers instant relief, while the tender white flesh delivers a subtle sweetness that has sustained generations. Austronesian seafarers, master navigators of the Neolithic era, carried these cultivated coconuts, including prized forms such as the compact Fiji dwarf, Niuleka, on their epic migrations across the vast Pacific in outrigger canoes laden with the promise of future groves. Genetic echoes even reached the Pacific coasts of the Americas long before European contact, preserved in ancient DNA signatures and subtle archaeological traces of early cultivation. In contrast, the Indo-Atlantic group emerged along the southern shores of the Indian subcontinent, including the palm-fringed beaches of Sri Lanka and the Maldives, where the air is thick with the briny tang of the sea and the distant calls of seabirds. Cultivated independently by ancient Dravidian societies, these coconuts, often tall, cross-pollinating types with larger, more angular fruits, became staples of coastal life, their swaying silhouettes dotting the landscape as they spread westwards. While they generally lack the full suite of refined domestication traits present in their Pacific counterparts, the existence of these two separate centers of early cultivation highlights a fascinating pattern. Humans independently recognized and nurtured the coconut in different ocean basins. Yet the Pacific Southeast Asian lineage ultimately became the dominant force behind its global success, its fruits traveling farther and influencing more lives. These genetic revelations gain even greater depth when woven together with archaeological finds, ancient texts, and linguistic evidence. Coconuts did not merely float passively across the seas. They rode the waves of human history as vital companions. For Austronesian voyagers slicing through turquoise waters, the palm provided crisp drinking water that stayed fresh for weeks, nourishing flesh that could be eaten raw or dried into travel rations, and strong fibers twisted into ropes and sails that whispered reliability with every gust of wind. Trade routes carried Pacific coconuts to Madagascar and the East African coast, where genetic admixture with local Indo-Atlantic populations still echoes those ancient exchanges in the mixed groves swaying under monsoon skies. Later, Arab merchants gliding along spice-laden winds, followed by European colonizers and the engines of global commerce, blended populations further, planting coconut groves that now fringe tropical shores from the Caribbean's turquoise bays to the warm currents of the Indian Ocean. The architecture of dwarfism. Hormone mutations. The transition from tall, wild-type palms soaring over 20 to 30 meters high to compact, early-bearing dwarf trees represents one of the most significant triumphs of ancient Southeast Asian selection. Understanding how a giant palm was transformed into a manageable, highly productive food source requires peering directly into the molecular mechanics of the plant's growth hormones, specifically the gibberellin, GA, pathway. Gibberellins are diterpenoid hormones that drive cell division and stem elongation. In tall coconuts, uninhibited gibberellin synthesis leads to long internodes, delayed flowering, often requiring 6 to 10 years or more, and massive vertical height. In dwarf varieties, mutations within this pathway interrupt normal stem elongation, yielding short internodes, a compact canopy, and precocious flowering within just 2 to 4 years of planting. Genomic sequencing of tall and dwarf cultivars has isolated 2 major regulatory hubs responsible for this morphological change. 1. GA20-oxio-X, biosynthetic gene regulation. The enzyme GA20-oxidase catalyzes the rate-limiting steps in producing bioactive gibberellin. Rather than a total knockout mutation, which would render the palm sterile or severely stunted, dwarf coconuts possess a refined set of quantitative genetic modifications. Copy number variation, CNV. Tall coconut genomes carry greater copy numbers of key GA20-ox genes, typically up to 5 functional copies, compared with dwarf genomes, which frequently carry only 3. This reduced gene dosage directly lowers the overall volume of synthesized growth hormone. Promoter single nucleotide polymorphisms, SNPs. High-throughput sequencing reveals a crucial single nucleotide change in the promoter region upstream of the GA20-ox gene on chromosome 12, locus GZ to G04578. This variation significantly suppresses promoter activity in dwarf tissues, lowering gene expression by anywhere from 1.3-fold to more than 12-fold compared with tall palms. CNGA20-OX1 versus CNGA20-OX2. While coding sequences for CNGA20-OX1 are largely identical between tall and dwarf variants, expression levels of CNGA20-OX1 in leaf and stem tissues are substantially higher in tall palms. CNGA20-OX2 remains low or tissue-specific in both, indicating that regulation occurs primarily through promoter elements and gene dosage rather than structural protein disruption. 2. DELA signal repressors and growth regulators. Downstream of hormone synthesis lies the signaling mechanism governed by DELA proteins, such as CN-DELA1, and growth-regulating factors, such as CNGRF1. DELA proteins act as master negative regulators of growth. When gibberellin levels are high, DELA proteins are targeted for degradation, allowing the plant to grow tall. When gibberellin levels are low, DELA proteins accumulate and suppress growth. Targeted PacBio long-read sequencing across diverse coconut populations has revealed key genetic variants within CN-DELA1, including specific deletion patterns and diagnostic markers, such as MN-DELA1 score SNP1270 and MCN-GRF1 672. These markers correlate strongly with the dwarf phenotype, modulating the palm's sensitivity to available gibberellins and reinforcing the compact growth habit. Together, reduced GA synthesis and altered DELA repressor dynamics create a balanced, highly productive dwarf tree. 2 distinct dwarf paradigms. As human migration and trade carried coconuts across different regions, selection pressures created distinct dwarf varieties suited to local environments and human needs. The Malayan dwarf of maritime Southeast Asia and the Chowgat green dwarf of southern India represent 2 of the most prominent, well-characterized dwarf varieties in modern agriculture. The Malayan dwarf, the Pacific paragon. Originating in the heart of the Pacific lineage in maritime Southeast Asia, the Malayan dwarf, occurring in distinct color forms, green, yellow, red, and golden, is a premier example of human-driven domestication. Driven by a mutation enabling strict autogamy, self-pollination due to overlapping male and female flowering phases, the Malayan dwarf achieved rapid genetic fixation. SSR marker studies show high levels of genetic uniformity within these color lines. The tree reaches a modest 4 to 10 meters, bearing vivid clusters of rounded, high endosperm fruits close to the ground. Aside from its early yield in sweet water, the Malayan dwarf gained global agricultural prominence because of its natural resistance to lethal yellowing disease, a phytoplasma disease that decimated tall coconut plantations across the Americas and the Caribbean. The Chowgat green dwarf, the Indian sentinel. Hailing from the Chavakad region of Kerala, India, the Chowgat green dwarf, CGD, alongside its orange variant, represents a parallel outcome within the Indo-Atlantic sphere. Reaching 4 to 5 meters in height and flowering within 3 to 4 years, CGD also exhibits nearly 100% self-pollination, resulting in remarkable phenotypic and genomic homogeneity across generations. Chromosome-level assemblies of the CGD genome, approximately 2.68 gigabytes, have revealed more than 300 resistance gene analogs, RGAs. Unlike the Malayan dwarf's reputation for lethal yellowing resistance, CGD was selected for its exceptional tolerance to root wilt disease, a devastating condition endemic to southern India. Its medium-sized, bright green nuts remain a staple for tender coconut consumption across the subcontinent. The coevolution of humanity and the tree of life. Despite centuries of movement, interplanting, and commercial hybridization, the genetic fingerprint of those original Southeast Asian domestications remains strikingly visible in coconut populations worldwide. This persistence underscores a profound truth. While the coconut is a marvel of natural adaptation, its buoyant, salt-tolerant fruits perfectly suited to coastal life, enduring sun-scorched beaches and storm-tossed seas, it is also a deeply human plant, co-evolved with the peoples who cherished its shade, sustenance, and versatility. More than the biography of a single species, the coconut story offers a window into the intricate interaction between humanity and the natural world. From the coastlines of island Southeast Asia, where its domestication first flourished amid the perfume of frangipani and the distant roll of thunder, the coconut has reached tables, economies, and ecosystems around the globe. Split open on a market stall, its aroma evokes distant islands. Pressed into oil, it lights lamps and soothes skin. Woven into daily life, it supports entire communities. Today, chromosome-level reference genomes, transcriptomic analyses, and marker-assisted selection allow breeders to pair the compact stature and disease resistance of Pacific dwarfs with the rugged vigor of ancient tall lineages. Whether through the lens of evolutionary genomics, archaeological discovery, or the simple pleasure of tasting cool coconut water on a parched tongue, Cocos nucifera stands as a living testament to how human curiosity and plant genetics intertwined to shape the tropical world.